Hi, my name is Lucy Pavlovich, and I would be talking about string theory, but I think that's too much to talk about in a 10-minute video. So instead, I'm going to talk about robots. Well, not so much killer robots that are going to take over the world like you see in sci-fi books and movies, but rather a bioengineering innovation. As of now, there is a robotic arm that can be controlled by thought using a chip implanted into the brain. There's a way a robotic arm can be attached to the patient without having to go through the brain. The problem is that this would only work if you could do the procedure right when the person's limb is amputated, because it requires connecting nerve endings to other tissues in the body and connecting the chips there. However, this is impossible a lot of the times because people will need emergency amputations, or if they get caught in a bomb explosion, that isn't exactly the pristine lab procedure to ensure this reconnection. So, I developed a theoretical process in which a chip could be implanted into newly generated muscle in the arm and allows you to remove the robotic arm. This, theoretically, would work for anyone who has had an amputated limb, either just now or years ago. How, you may ask? Well, let me explain. When a person has a limb amputated, the motor neurons and sensory neurons are cut in half and begin to degenerate because there is no sensational feedback. We are not starfish. We cannot just grow back limbs. So instead, one concept is to take the person's skin cell and revert it back to a cell similar to stem cells using a process of IPS, induced pluripotent stem cells. 2012 Nobel Prize winner Shinya Yamanaka developed this process. This process could be revolutionary to the field of regenerative medicine because they can divide into cells any cell needed, such as neurons, heart cells, pancreatic cells, or liver cells. These IPS cells act like embryonic cells. They replicate and can be told to form into any type of cell based on the environment you put them in. In theory, once the skin cells are reverted back to stem cells, they can be separated and cultured into two groups, motor neurons and muscle tissue, by using specific chemicals. The motor neurons will be told to grow only into that one specific neuron, and the muscle tissue will be cultured around electronic impulse sensors. This allows the neuron to communicate with the robotic arm. Electrical impulse sensors work by measuring voltage fluctuations resulting from the ionic current within the neuron. Neurons are actually a fascinating thing. They are the only cell that allows electricity to run through them. They do this by polarizing and depolarizing every thousandth of a second. Think about it. Right now I am talking. Every time my mouth and tongue move, motor neurons are firing and shocking our muscles to do what they want them to do. How does it do this? Well, a message is sent via neurotransmitters, chemicals, to tell the neuron to start firing. The dendrites, receiving end of the neuron, has receptors to catch these neurotransmitters so that they know what to do. Then they start firing. Electricity flows down the axon by being propelled by the imbalance of ions on the outside and the inside of the axon. Once it has reached the end, the electrical impulse tells the end of the axon to release neurotransmitters, and the process starts over again with the next. These electrical signals are recorded and interpreted into different movement that are then reenacted by the robotic arm. After the motor neurons are cultured around the sensor, an amputee would get a stem cell injection, allowing them to regrow the motor neuron. Along with that, the new muscle tissue that has been cultured around the sensor will be added at the end of the person's amputation, and the neurons grow to connect itself to that. Hopefully this process will connect the spinal cord to the new muscle tissue. Then, when the person's body sends the signal through the neuron, the sensors will pick up the electrical impulse and work the robotic arm. Programming deals with the rest. The sensor reading is analyzed and converted into movement in the robotic arm depending on where the electrical impulse was, which sensor was detected, and the amount of electricity sent and the time lapse between impulses. If, for example, sensor 1 detects a signal, then the thumb will move. The robotic arm will know how much to move and how quickly to move based on the amount and time lapse. This could be a revolutionary idea, and I find it very interesting, and I hope that this video has helped to explain this concept, or even better, has gotten you interested in the field of bioengineering. Thanks for watching.